I'm Dr. Ben Morrill. I'm very happy to welcome you to the very first Reptile Genetic Counseling Podcast, Episode 1. I'm here with Kayla Short. She's going to be helping run things and asking questions.
Hey there.
And so the main idea that I have in doing this each week is to provide an opportunity to give all of you updates on what's going on. Since a lot of these genetic tests are brand new or very new, and many of you may be new to the genetic testing we're doing in reptiles. Just to provide an opportunity, we'll post each week in our social media some of the ideas, what we're going to cover, but also just have an opportunity for people to post questions there, and we can go to those questions and answer those in each episode. And then also, one of the first things, at least to start out with, that I plan to hit is talk about people who samples. All of you that are watching this that already sent samples in, we're wondering, when you're going to be able to get results. So we'll go ahead and go over that real quick. So right now we have samples that were collected over the last couple of weeks. By the time you see this, it was probably 2.5 to 3 weeks. So if you sent your sample in during that time, excluding the last 4 or 5 days, because those will go on the next run. So if your shed was received before about April 4th, and you don't have results yet, then your shed will likely be on this run. And that's whether it's sex determination or ball python morph testing. Those are going to into process over the weekend of April 8th and 9th, and we'll go on to a run next week. So by the end of next week, so I guess that'd be April 14th would be Friday, we should have results by then, and maybe even a day or two earlier if we're lucky, but yeah, it should be by next Friday. So I'll give you a little heads up, and we'll try to do that at the beginning if you want to be able to just pop in here and see when the next run's going to come. Sometimes I'll say it's still 2 weeks out. Sometimes I'll be able to tell you, yeah, you'll get it next week. But we do see as we move into the breeding season, we'll have a lot more sheds coming in. And so the way that it works for us to be able to keep the costs as efficient as possible for everybody, we have to batch samples. And so we have to have so many samples for a run. And so right now, where there's not as many people hatching babies out, we're a little early for the 2022 season. Our turnaround time has been closer to 3 weeks. But as we go into the summer, we'll have lots more sheds coming in every week. And so I would expect the turnaround times to go down. And I hope that there will be times where once we receive the shed, obviously we can't affect how long it takes to get to us. We can't speed up the mail. But yeah, once the shed gets to us, we should be able to, during the busiest time of the summer, I would imagine we'll get rundowns down to like a week to 2 weeks. So we're looking forward to that. We'll have lots of samples going through and be able to get results back to people quickly. And we are working with Clutch and Morph Market on ideas to be able to keep that flow up during the part of the year where we don't have as many sheds coming in from the baby season. As long as we can keep the number of tests coming in each week high enough, then hopefully that turnaround time can be more like 2 weeks and maybe even less instead of 3 weeks and sometimes more. So it really is dependent on you all sending samples in. The more that come in, the faster we can get another run going and get results out. So that's where we are with that. I think you have a question for me, Kayla.
Yeah, we sure do. So this is from Kyle Jones, I believe, on Facebook. And he's asking, what testing for ball pythons is available now?
All right. So Kayla got us a nice layout set up so that we can have this on the screen. And so right now, what we have is clown, piebald, lavender albino, hypo or ghost, candy or toffee. So those are hypo ghost, those are identical. Candy toffee, those are identical from the genetic work that Dr. Seidel at Eastern Michigan University did with hypo and ghost. That includes orange ghost. Those have all been identical in her testing and the testing that we've done as well. The hypo and ghost is the newest test we've had available. And so we've done over 100 sheds. She's done, several herself, and hypo, ghost, orange ghost, all of those have been identical to this point. But if you have a line that you think may be different, or you've tried to crossbreed with a hypo or a ghost in the past and didn't get hypomelanistic animals, we'd be very interested in running those and seeing if we can find something different. But so far, everything's been identical. And then with candy and toffee, it's the same thing. When Dr. Seidel did that work with her students there at Eastern Michigan, they found that candy and toffee are identical. And then we have albino and yellow belly, spark, specter, asphalt, and gravel. So all of those are available right now. And then in the future, and when I say available, at Morph Market and at Clutch. So both of them are gracious vendors that are helping to. I'm a scientist, and I'm not the best with electrical, all of this kind of stuff that Kayla's helping me with today. But yeah, as far as making things available in as easy and quick manner, and be able to keep the data in a all well put together and make it quick and easy for us to get results out once we do get the work done. We're very thankful to Morph Market and Clutch for that. So you can go to either of their genetic testing buttons and find our tests available there. And then for the ones that are coming up, I know people are always asking about this. So Ultramel, we did have available for a little while. I was able to, for those of you that don't know, with Ultramel, there's actually at least 2 different mutations that cause Ultramel. So similar to albino and yellow belly, there are multiple mutations that cause that morph, that phenotype, what we're seeing, or a trait would be another way to call it. So what we're seeing as an Ultramel can be caused by 2 different genetic mutations. So one of them we can very easily test for, and it's the more common of the 2. So some of you will have sent samples in before, and we gave you a result that it is Het Ultramel. That is true. That was a good result. If you got a result back that said negative for one allele or negative for one mutation, that's what we meant. For the one that we could test for, it was negative, but there is still a chance that less common Ultramel mutation could be in that animal, but we are blind to it right now. I had a test that was picking it up some of the time, but not a high enough amount. We want our test to be over 99% accurate, and so it's not high enough for us to make Ultramel available again. But we are actively working on it. Dr. Seidel is actively working on it. So as soon as we have that other mutation where we can test for it and we can see it, we'll make Ultramel available again. And then Desert Ghost, we've been working on for a long time. I know many of you have asked many times, and I'm sorry it's taken longer than normal. It's been a difficult, much more difficult one to get than Clown was. So Clown is one that we found, we mapped and designed. Desert Ghost is another one that we've been working on. Hypo Ghost that Dr. Seidel got, we did help some. We provided some whole genome sequencing data to help kind of check and make sure there weren't any other mutations like we were just talking about with Ultramel that maybe would cause it. So we helped some with Ghost and Hypo, but the one that was ours was Clown that we mapped, and then Desert Ghost is the second one. So we do have a Desert Ghost test that is, if I get a negative result, so far I'm just running known samples. So that's part of the process designing a test, is I have to have a bunch of known samples. I know whether it's no genetic or no Desert Ghost at all, if it's a Het Desert Ghost, or if it is a visual Desert Ghost. So I have about 150 where I know for sure what their genotype is, whether they're supposed to be negative, Het, or homozygous or visual. And so right now with Desert Ghost, the test that I have If I get a negative, that's right 97% of the time. So that's great. That's almost to 99%. But the downside is for the ones that it calls het, it's only right 85% of the time. So we're very close. We know we're in the right region. There's other mutations that I'm going to keep testing over the next week or 2 or 3. I'm hopeful that the next iteration will improve it to a point where we can make that available. So wouldn't surprise me at all within the month if we have Desert Ghost going. And sometimes things just end up being more difficult than we thought. It's an iterative process. We'll get to a point where we're ready to run a bunch of known samples, and then we find a problem. We have to kind of go back to the beginning, redesign, test a few, maybe we'll test 3 or 4, and then we'll test a dozen, and then we'll test 50, and then we'll test 100. And so it kind of has to get through all of those phases for us to say, okay, it's over 99% accurate. We're ready to make it available. So Desert Ghost is getting really close. Genetic Stripe, we're collaborating with Dr. Siddell directly for Genetic Stripe. And in working with her, we have it, some of the information from us, some of the information from her, we're able to figure out what gene is highly likely. We don't know for sure yet, but we do have a gene that is highly likely the gene that is mutated for Genetic Stripe. And so now we are in the process of going through base changes or mutations that we see in Genetic Stripe samples and figuring out which one is the correct one that's only present in Genetic Stripes or had Genetic Stripes and not present in the ones that do not have Genetic Stripe or should be negative. So we're in the process of that. So Genetic Stripe, that's probably a month or 2 out, but it's one of the closer ones. And then the ones we have in sequencing right now, so we get dozens of samples of Sunset, in this case, Sunset, Monarch, VPI Axanthic, and TSK Axanthic. So we have dozens and dozens of samples that we will get the whole genome sequenced. It's called low-pass whole genome sequencing. And so we'll get a lot of information on those dozens of animals that are either Sunset, het Sunset, or negative. And we have probably 300 or more now that we've done that with that are negative for Sunset. So we have a pretty good group to be able to compare. And then we are working to do comparisons with those genomes to find what the base change is for Sunset, and then what it is for Monarch. And then obviously, and the Axanthics. And then obviously we'll have to figure out if maybe there's more than one mutation causing it, and that makes it more complicated and more time for us to figure it out. But so Sunset, Monarch, VPI Axanthic, TSK Axanthic, I would say those we're hopeful that sometime in the summer, by the end of the summer, we'll be able to add at least some of those. And if we get lucky, all 4 of them. All right. So the next thing that we want to cover is sex determination tests. And so for sex determination tests, as many of you may know, we have been testing Colubrids for sex determination from shed skins for 3 or 4 years now. I think maybe it is even a little over 4 years. So that's our main sex determination test that we've been running long term. We also more recently have a ball python sex determination test, and that's working very well. And then ones that we're working on. So in the very beginning, when Sean Christian, so Sean is my business partner with Rare Genetics Inc. He does lots of work in the background, and many of you, if you've asked questions or called in, you've probably talked with Sean or messaged back and forth with Sean. So Rare Genetics Inc. really started in 2011 when Sean and I met in person. We'd been friends online for a while, but we met in person at a Tinley Reptile show in 2011. And really the first thing that we were really interested in being able to do would be a sex determination test for green tree pythons. And so I guess right now would be a good time, Kayla, if you want to read the question that we have about that.
Yeah, sure thing. So this was from Jason Borges. I hope I'm pronouncing that right. He asked, when can we expect single shed testing for chondros to be available?
So to answer your question, Jason, probably longer than you expected, but so this is the test we've literally worked on the most. We started with this. I can't express to you how frustrated I've been many nights. And I know there are many green tree python keepers that have been frustrated as well. We've spent a lot of time and a lot of money. For a little while, I figured out a way that I could do some sequencing on the sex chromosomes and use information, ancestral information. So keepers had to send in a shed from like the sire, whatever like say you wanted a clutch of green trees sexed that you just hatched out, the breeders would have to send in the shed from the sire, and then also either the grandsire, the paternal grandsire, or a known son from the same sire, so that I could follow that sex chromosome and be able to see markers on it. And so it was very analysis heavy, and I definitely was making no money at it. But it was something I really wanted, something that we could have that worked for green tree python keepers. So I did do that for a while, and that did work. Haven't had anyone come back and say there was a problem with that. So that was really cool. For sure, know that the markers I was looking at are on the sex chromosome, did pass from the sire to his male offspring, and then any that were offspring from him that didn't get those markers, I knew were the females. But when we switched and we scaled up to where we are now, it changed the way the type of samples that we're running. And so I have to have a big enough batch of samples to be able to make it be cost effective. And so it made it so I couldn't do the green tree python test how I used to, but I had information from the ball python and that sex determination test, and I thought I was going to be able to fairly easily figure out there in that same site or somewhere close to it, so that we would have a green tree python test. And so I tried over and over many things, and knowing from what I did with the Colubrids, I was able to move through different species and genera within Colubrids and figure out tests that would work in general over large unrelated members of the Colubrid group. But I have not been able to do that with green tree pythons or other pythons. And so that those sex chromosomes seem to be different enough that's been complicated, unfortunately. So you picked a very deep question to ask, and you may not have known that, but it's definitely something that both Sean and I really want to get working. We are sending more known male and female green tree python sheds to get sequenced so that we can do that comparison, more comparisons. I don't know for sure if the sex determining region is different in green trees than it is in ball pythons, or if it's just so variable between the two. They're from different genera. The green trees are in Morellia, whereas the ball pythons are in Python genus. I don't know if it's just really variable, or if that region is on a different part of the sex chromosome. I thought it was going to be a lot easier than it's been, but this next batch of sequencing that we're doing would kind of follow what we were just talking about with the Axanthics and the Sunset and Monarch. So I'm hoping sometime in the summer we would have green tree, and I'm still trying some things on the data I already have, which I've been failing so far. So there's a small chance that I could get lucky with something that I keep trying, that I find something that will work, but I just don't know for sure. I'm very hopeful, and you can rest assured we're working on it. Other people have attempted to design a sex determination test for green trees, and there is none available by anyone else either. So I think other people have found it to be difficult as well. But yeah, we are working on it, and we are hopeful that sometime this summer, by the end of the summer, I sure hope we have it by then, and I'm still crossing fingers. Maybe we can get lucky before that, but we'll see. Other ones species that we're really interested in doing and have made some progress on would be blue-tongued skinks. So many people have asked me about that as well, and I've talked about it for a long time, but we have made progress. It's not ready yet. That's another one that could be sometime this summer where we can start to do blue-tongued skinks. We're interested in many others, but that's the other one that's close. All right, Kayla, where are we at now?
Let's see. Next, we have identity testing.
Oh yeah, so identity testing. This is a fun topic. So there are some groups that have contacted me, and we've worked actually over the last year and a half, excuse me, to develop some tests that incorporate the whole genome. So this low pass whole genome sequencing we're doing to be able to find new mutations. We can also do that in an individual, and that will give you a unique genetic identity for that individual. You could tell it apart from its siblings. This kind of information has been used in scientific literature for wild animals to be able to tell, how closely related individuals are, for paternity testing. It would certainly give us something that we could use and it'll take a little time to bank information, but to be able to tell purity, to be able to tell locality. So there's a lot of species of reptiles that people keep that they want to know if it's a cross between two species, or they want to know if it's a certain locality. Madagascar boas, we've got some good progress on that to be able to do identity testing there, be able to tell if it's a cross between the Madagascar ground boas and the Dumerals. So that's one of them I've been working on the longest. And Panther Chameleons is another one. And we have that's in sequencing, that's going as well. Another one that will be very helpful to indigo keepers, we are working on an identity test, and that something similar has been available, looking at, I think, 10 or 15 markers to be able to tell individuals apart. The identity test we've made the most progress with, just because we have the most sheds, is the ball python one. And so with that one, we were able to find 10,000 markers, variable markers that are coming up in these low pass whole genome sequencing runs. So you can imagine that the power that we can have if we know at 10,000 different locations, an individual, whether they have an A or a T or a C or a G there, we would know whether that means it's, from a certain locality, or if it's a cross, or if, like I said, paternity testing will be able to tell for sure who the sire is. And if people had a question, hey, I got this green tree python, is this really the lineage from such and such, blue line or whatever, we'd be able to have genetic proof that that is indeed from that lineage. So there's a lot of really cool things that we'll be able to do. And it's something that over time, as we have more and more sheds coming in, people running this, that we'll be able to say more and more. As a PhD student, I worked in cattle reproduction, and there they have what they call EPDs, or estimated progeny differences. And with that, they're able to tell like a certain bull, they can tell whether the offspring, if it's for milk cattle, they can tell which bull would be the best one to use to have the highest milk output from a female offspring from that bull. So that's just one small example, but that's something we could potentially build a database and figure out, which females are the best ones to keep for large clutch size, which sires produce female offspring that tend to have more healthy offspring per clutch, or to lay more often, or, there's several questions. We can also start to follow longevity. There's a lot of questions people have about longevity and what they feed. Well, another thing that you can ask is, are there genetic reasons why some are more prone to RIs or egg binding, which in ball pythons isn't as big of a problem as other species. But there are a lot of things that, and actually for my dissertation, I worked with the snake keeper with Dan and Colette Sutherland and looked at, I think it was about 10,000 eggs over 10 years. And we're able to calculate heritabilities for some of those reproductive traits, that they are in fact heritable from one generation to the next. So that's another area that with this identity testing over time, as we have more sheds come in and more information, that we'll be able to do some really cool things, be able to know more about the animals and make better selection, for if you have 3 or 4 different males that have the same genes, if it's a confusion, lavender albino clown male, you've got 3 of them you hatched out, you want to know which one you want to keep. This type of an identity test would tell you which one has is the most outbred, or the least amount of inbreeding depression, would be more likely to have female offspring that have large clutch sizes, whatever the selection criteria are. This would give us a genetic way to make our choices of which animals we're going to keep and breed. So I'm pretty excited about it. It'll take some time, but we're making the first steps in developing these tests for doing the identity testing. And even at first, if it's just to have an identity, if you wondered later, hey, is this really an animal that such and such had, you could just send a shed in and run that identity test again and say, yes, that's identical, or, no. So just as an identity, instead of putting a chip under the skin like they do in some animals, just at any point you can send a shed in and we can say, yes, that is this specific individual. And we would have very, very high probability match, since we have so many locations where we have variable markers. So I'm pretty excited about that, and to see how that grows over time. All right, Kayla, was there anything else?
I believe that's it. Let's see. Do we have any final thoughts for the topics that we've covered today?
I just want to say thank you for watching. If you made it to this point, I know I probably get a little long and rambling about some things. This is stuff I've literally been thinking about since 2011, pretty much every single day. I'm very, very thankful to all of you who have sent in sheds, especially when we were really small, just doing Colubrids sex determination. I had a big uphill to climb, and also very thankful to Sean for having, the capacity to support and to be patient as things slowly come along. I've got 4 kids. I got a PhD, then I moved across the country and got a full-time job. So it's been nights and weekends for many, many years. But this year I was able to quit my full-time job, and this is what I'm doing 100% now. And so
All right.
It's very heartfelt, thankful. Thank you to everybody, Morph Market and Clutch, for helping us make it available and this possibility known to lots of people. It's been a really, really cool thing to see grow, and I'm very excited to be able to run these tests and develop new ones, and be able to talk with you all more, and over as we do follow-up episodes from this one, we'll integrate some pictures and videos, and we'll be able to tell some of the stories. There's some cool stories of things people have found out, and there's one that I just saw today where someone did a panel to be able to tell yellow belly or gravel, but they figured they'd just spend some more and do the whole panel. Found out they had a het pied. And so that het pied, he got in touch with the breeder that he bought it from and found out, oh yeah, 2 generations back, there was a really small chance that there was some het pied in there. And so now that breeder has some cool clown projects that he has going on that are all pos het pied. So yeah, it's pretty cool. I don't know if you have that handy Kayla the name of that person. If not, I'll have it.
Yeah. Hang on. I think I've got that. That was
Let's see. Oh, I've got it. George
Yep.
Longfellow. So yeah, thank you, George, for sending that in to me. Those kinds of stories are what feed me. If I'm up till 2 or 3 in the morning designing something new or stuffing sheds into little tiny vials or whatever, I love those stories. So we'll share some of those stories on here too. Thank you
Definitely.
so much. And I think that's it, Kayla.
Awesome. Right, and thank you, Ben, for sharing so much. I think I speak for the audience when I say that we've learned a lot just from your work today. So everybody, make sure to let us know what you think. Give us questions for future podcasts, and I think that's a wrap.
Thank you.
Thank you, guys.